Dr. Laurel Kartchner, and I'm here today to talk to you about an article that was recently published in Kidney International titled "A Mendelian Randomization Study Found Causal Linkage Between Telomere Attrition and Chronic Kidney Disease." Chronic kidney disease is a condition in which the kidneys have experienced some sort of damage. There's a wide variety of causes of chronic kidney disease. For example, hypertension can cause chronic kidney disease, diabetes can cause chronic kidney disease, and genetic factors can cause chronic kidney disease. Now, chronic kidney disease has been shown to be highly prevalent in an older population, in older individuals. Older individuals have also been shown to experience something called telomere attrition. So telomere attrition essentially relates to chromosomes that are present in the cells. So DNA is packaged in chromosomes. It's tightly packaged into chromosomes that are present in the nucleus of a cell. And at the end of each chromosome is what's called a telomere. This is essentially a DNA sequence that acts as a sort of cap to protect the DNA from being degraded by the cellular machinery around it. So by having that telomere, you're able to protect the DNA. There are a number of conditions that have shown that when you shorten that telomere, it can cause some sort of degenerative disorder. So for example, this has been shown in coronary artery disease. It's also been shown in Alzheimer's. So this begs the question of, since we know that telomere attrition happens with age, so you get the shortening of that telomere, and we know that telomere shortening has been associated with these degenerative disorders, is there a linkage between the telomere attrition that happens with age and this highly prevalent chronic kidney disease that takes place in older individuals? There's a technique that can be utilized to kind of interrogate this question called Mendelian randomization. So Mendelian randomization is a technique that allows you to reveal whether or not there are causative effects between different factors that you've observed. And so it basically lets you predict whether or not there's a causative effect from these predictable exposures in these really complex diseases. It lets you tease out the potential relationship between two factors. In this study, they performed two sample Mendelian randomization analysis. They wanted to test the causal linkage between telomere attrition and chronic kidney disease utilizing two population-scale independent genetic databases. They hypothesized that a shorter telomere length would be a causal factor in increasing the risk of chronic kidney disease. They also investigated the reverse direction causal effects of kidney function impairment on telomere attrition. So they looked both directions in this analysis. Mendelian randomization requires three main assumptions about the instrument being utilized. One is that it's going to be strongly associated with the exposure phenotype, that it's not associated with confounders, and they have a number of ways to verify that and to confirm that. And the third is that causal effects should be through the exposure trait. This third one is not actually testable, but utilizing a median-based Mendelian randomization can kind of relax this assumption. They also introduced the fact that telomere length was introduced. Examining telomere length was introduced in a previous GWAS meta-analysis for leukocyte telomere length in another study. The group utilized two large population-based genetic databases in order to determine what SNPs should be utilized to test for telomere attrition and what SNPs should be utilized to test for kidney function in order to generate the instruments needed to examine both directions the effects of telomere attrition and kidney function. They were able to create a summary level Mendelian randomization for both the telomere attrition and the kidney function, and they were able to create a polygenic score-based Mendelian randomization for the genetic instrument for telomere attrition. Utilizing the SNPs that the authors identified in their initial analysis, they found that there was a causal relationship between telomere attrition and CKD stage 3 or higher. In fact, they found that there was an odds ratio of 1.2, which was found to be a significant difference. And when you further exclude variants that were shown to have a weak association with telomere length, but were strongly associated with other phenotypes, there is still an increased odds ratio showing that there is a potential causal estimate, a causal relationship between telomere attrition and CKD. Examining the polygenic score analysis results, the authors found that there remained a causal relationship between telomere attrition and CKD when examined with the polygenic score system. When you adjust the model for various clinical covariates, they found that this increased odds ratio remained and was also statistically significant. The authors also decided to look in the reverse direction to determine whether or not kidney function impairment would be causative of telomere attrition. And they found that there was a significant causal effect between kidney function impairment and telomere attrition, as shown by a beta score of 0.39, which was found to be statistically significant. When they excluded SNPs that were strongly associated with potential confounders, they found that that beta value was still 0.33 and that this was still statistically significant. So in summary, what the authors found was that there was a causal relationship between telomere attrition and chronic kidney disease. And they looked at both a summary level Mendelian randomization and an individual level or polygenic score based Mendelian randomization. And they found that this relationship held true in both cases. And they also found that there was a causal relationship between kidney function decrease and telomere attrition as well. This study utilized two genetic databases that, while broad, largely consisted of individuals of European ancestry. As such, it will not reflect the demographics of a broader world population. Additionally, while the authors utilize Mendelian randomization to determine that there is a potential causal link between telomere attrition and chronic kidney disease, they do not discuss potential mechanisms or propose any sort of scientific studies that can be utilized to confirm this finding as well. In summary, the authors utilize Mendelian randomization to determine that telomere attrition likely is a causal factor for chronic kidney disease, and that chronic kidney disease may also in turn affect telomere attrition as well. Thank you for your time, and I hope you have a great day.